Good morning. I am Dr. Anand, Associate Professor in Department of Anatomy, the Lakshmanarayana Institute of Medical Sciences. So today we'll be seeing about the histology of cartilage. Okay. So what is a cartilage? So it is a type of tissue. Okay, it is a type of tissue. So there are three types of cartilage. So you'll be able to have hyaline cartilage, elastic cartilage, white fiber cartilage. So these are the major three types of cartilage you'll be able to have. So for example, for hyaline cartilage is your trachea, white fiber cartilage is your intervertebral disc or your costal cartilage. For your elastic cartilage, it is your pinna or epiglottis. Okay, so these are the structure that are example for the cartilage. Okay, so now we will see about the component of a cartilage. So what are the things you would be able to see a component in a cartilage or cells? So you would be able to have cells. So these are nothing but your chondrocytes. Okay, so those are going to form the cells of a cartilage. For example, in a muscle, you would be able to get the myocytes. In case of bone, you would be able to get osteocytes. So like that here, you would be able to get the chondrocytes. Okay, so these cells are developed from your mesenchymal tissue. So from the mesenchymal tissue, you will be able to get your chondroblast. So these chondroblasts are going to get converted into chondrocytes. Okay, so these cells are going to helpful in the formation of your cartilage as well as maintenance of your cartilage. Next thing, you would be able to get your matrix or ground substance. So this is nothing but the area in between the cells. Okay, so it is made up of glycoprotein structures. Okay, so this is the another important thing. Next, you would be able to get your fibers. Okay, so you would be able to have different types of collagen fibers. So here you would be able to have some fibers. Okay, so these are the three main component of a cartilage. Okay, so the cells are made up of chondrocytes and chondroblast, and the matrix, it is made up of ground substances. So what are the ground substances it is made up of? It is made up of protein and carbohydrates. The carbohydrate is mainly made up of glycoprotein, glycoaminoglycan. Okay, glycoaminoglycan, and the proteins are made up of chondroitin sulfate, kerosene sulfate, and hyaluronic acid. So these are the structures that are going to contribute for the formation of your cartilage, and the core protein is your aggregan. Okay, so next, as you see the hyaline cartilage, so this is the histological structure of a hyaline cartilage. So in the histological structure of a hyaline cartilage, you will be able to get the following structure. So we are seeing that you have three important things in a cartilage. The first one is your chondrocyte, that is the cell. So here you will be able to get your chondrocyte. So these are your chondrocyte. So next you will be able to get your proteins. Okay, so here you will be able to get, this is your extracellular matrix, so which is made up of your proteins, that is your proteoglycan and collagen fibers. Okay, so here in between the matrix here, you will be able to get your collagen fibers. So you will be able to have different types of collagen fibers. So normally you will be able to have type 1 and type 2 type of collagen fibers in the cartilage. Okay, so here you would be able to get the fibers, but normally these fibers are not able to be seen in case of hyaline cartilage. So this is because of the same refractive index of the hyaline cartilage, that is a matrix of the cartilage and the fiber. Okay, so they are going to have same refractive index. So because of the presence of same refractive index, the fibers are not visible in the slides. Okay, so you will be able to get only the, if you are able to observe, you will be able to only the matrix and the perichondrium, sorry, chondrocytes. Okay, perichondrium and the chondrocytes. So you cannot be able to visualize your collagen fibers. Okay, so in case of other epithelial cells, sorry, in case of other cartilages, elastic cartilage and in white fiber cartilage, there you would be able to observe your collagen fibers. Okay, here you can visualize by doing special stains. Okay, you can visualize the fibers by using special stains. Okay, so here you will be able to see. So this is a cut section of your cartilage. So on either side of a cartilage, you would be able to get the perichondrium. So in the bone, you would be able to get the periosteum. Like that, this is the outer covering of the cartilage. So this is called as the perichondrium. So below the perichondrium, you would be able to get your chondrocytes. So this is your, here you would be able to get your matrix. Okay, so this is the matrix. So in between the matrix, that is in the ground substance, you will be able to get your chondrocytes. Okay, so these chondrocytes are packed, or they have been situated in different types of things. So you'll be able to have 2 cells or 4 cells or 3 cells. Okay, they are being closely packed. Okay, so in a normal slide, you won't be able to see this thing. Instead of that, here you would be able to get the lacunae only. Okay, you would be able to get the lacunae surrounded by the chondrocytes. So this lacunae is being formed because of the sinkage of the chondrocyte during the process or during the tissue processing for staining procedure. Okay, so at that time it is going to get contracted. So there you would be able to get the lacuna. Okay, so this is another important thing. Okay, so here you would be able to get, so you would be able to get the perichondrium. So these are the outer aspect of the cell. So the chondrocyte, that is your cell, then you'll be able to get the ground substances. So these are the ground substances, and inside the ground substances you'll be able to get your collagen fibers. Okay, so normally the cartilages are very less nutritious. That is, the nutrition, it doesn't have any direct blood supply. So the blood supply is mainly due to the diffusion of nutrition from the surface towards the center of the cell. So how does the nutrition is going to diffuse? So from the perichondrium, the nutrition is going to pass and they are going to enter into the cell. So this is, so because of this, the healing nature of the perichondrium, that is the cartilage, is going to take much time. Okay, so since it doesn't have any direct blood supply, so what happens? The blood supply is not directly present, so it will take time to heal up. Normally, during the diseased condition and in case of accident, that is at case of rupture, so at that time, these cartilages are having very less amount of nutrition to heal faster. Okay, so why? Because of the diffused type of nutrition. Okay, so here this is another important thing. So these are the component of a cartilage. So you have the perichondrium, chondrocyte, then you will be able to get the matrix below outer, and then you will be able to get the type 1 collagen fibers. Okay, so how does this cells are going to grow? So here you will be able to get the cells are being packed closer. Okay, so here, as you see, the perichondrium, okay, which is present in the outer aspect, so they are going to have the chondroblast. So they are gradually secreting or depositing the new ground substances so that it is growing outwards in direction. Okay, so this is another important thing. So here you will be able to get different types of growth. So one is your interstitial growth. Okay, so here you would be able to get undifferentiated mesenchymal cells. Okay, so here the first thing here, this is your undifferentiated mesenchymal cells. So these are going to be getting differentiated. Okay, so these are going to be differentiated into or changed into chondroblast. So normally the mesenchymal cell is the stem cell, so it can take whatever the type it wants. So it can take change into osteoblast, it can change into a myoblast. So like that, it may go for any formation. So like that, the mesenchymal cells are going to form the chondroblast. Okay, after the formation of the chondroblast, so they are going to secrete more amount of extracellular matrix. Okay, they are going to secrete more amount of extracellular matrix and form the lacuna. So this is your extracellular matrix, this blue color line. So this is your extracellular matrix. So in between the cell, you are going to have a smaller gap. So those are called as the lacuna. Okay, so here these are called, now they are changed since they have secreted the extracellular matrix. So now they are grown into a bigger cell. So now they are called as chondrocyte. Okay, they are called as chondrocyte. So these chondrocytes start to divide into number of divisions. Okay, these chondrocytes start to divide into number of divisions. So they are going to form a grouping like structure. So these are called as isogenous grouping. Okay, these are called as isogenous grouping, otherwise called as cell nesting. Okay, these are called as the cell nesting. So now each chondrocyte lays down the matrix, causing to form the own lacuna and spread apart. Okay, so now each and every chondrocyte start to produce its own matrix, and they are going to move away from one another. So like this way, the cartilage is being growing. Okay, so this is one type of growth called as the interstitial growth. Okay, next you will be able to get the oppositional growth. So oppositional growth is nothing but the capillary force. So there the nutrition is going to diffuse. So as it diffuses, the chondroblast takes the nutrition and they are secreting new ground substances, and they are going to move in the opposite direction. Okay, they are moving towards the opposite direction. Okay, so like that it is another type of growth. So you'll be able to have interstitial growth and oppositional growth. Okay, so this is the cartilage. So this is a view of an hyaline cartilage. So this is the somatic diagram to show. So here you will be able to get your perichondrium. So here you will be able to get your chondroblast. So below that, the chondrocyte, they have started to secrete number of extracellular matrix. So in the extracellular matrix, you are able to get your chondrocyte. So this is your chondrocyte. The gap between the chondrocytes and the extracellular matrix. So this is called as the lacuna. Okay, so initially they are going to be present in two cells. They are dividing into two. Afterwards, what they are doing, they are going to get separated by secreting extracellular matrix. So in this way, they are going to grow. Okay, so this is a view of a hyaline cartilage. So here you would be able to get your matrix. So this is your matrix. So these are nothing but your lacuna. So here you would be able to get the chondrocytes. They are very small and they are undergone much shrinkage. Okay, so this is because of the procedure, staining procedure. Okay, so normally they are having lacuna, which has not been normally appreciated, but during the time of tissue processing, the lacuna gets larger. Okay, so this is your perichondrium. So this is your perichondrium. So here you will be able to have number of chondroblasts. So this is your hyaline cartilage, where you would be able to have number of chondrocytes, and this is your extracellular matrix. Okay, so this is to show you the same picture. So here you will be able to get the these are the cell nesting stage, where the cells are being arranged in 2 cell, 3 cell, or even in 4 cells. So here you would be able to see that you would be able to have 4 cells, 1, 2, 3, 4. Okay, and here you would be able to get 3 cells, 1, 2, 3. So it is based upon the multiplication of the cells. Okay, so here you would be able to get, this is your lacuna. So here you would be able to get the chondrocytes, and this is your matrix. So what are the identification feature for a cartilage, hyaline cartilage? So you would be able to have the perichondrium. Okay, so you would be able to have the perichondrium. So within the perichondrium, you would be able to get your chondroblast. So inside the cartilage, inside you would be able to get the matrix or ground substances filled with the chondrocytes. Okay, it has been filled with the chondrocytes. Okay, example for hyaline cartilage is your trachea. Example for the hyaline cartilage is trachea. Okay, so next, this is another picture to show you in 400x. Okay, how it is being seen. So this is your lacuna. So this is the cell. Okay, this is the cell, and this one is your ground substances. Okay, so here you will be able to get your pericardium. Okay, so this is to show you the same thing. Okay, this is the lacuna, and this one is your chondrocytes. So this is your matrix. Okay, cell matrix. So this is your perichondrium. This layer is your perichondrium. So here you will be able to get number of cells. So these are nothing but your chondroblast. Okay, these are nothing but your chondroblast. So next, coming towards an elastic cartilage. So we are seeing the hyaline cartilage. So that is the base for you. So now we will be seeing about the elastic cartilage. So in the elastic cartilage, what are the things you would be able to see? So the same thing here also you would be able to have the perichondrium, inner and outer layer. They are being formed with the perichondrium, and you would be able to get the same thing, that is the chondrocyte here, and surrounding the chondrocyte you would be able to get the lacuna. So in between the chondrocyte you would be able to get the ground substances and matrix. But here the thing is, the cell is being filled with elastic fiber. Okay, the fibers are being filled with elastic fibers. Okay, so there we are seeing type 1 and type 2 collagen fibers. Here it has been filled with elastic fibers. So because of this only, the cartilage are elastic in nature. For example, when you are going to squeeze your pinna, so what would be happening? It would be coming to its original position, or it would be retracting to its original position. So this is because of the presence of your elastic cartilage. But here the difference is the cell nesting is not present. The cells are individual. Okay, they are being present individually. Okay, so here also you would be able to get the chondrocytes, and at the periphery you would be able to get the chondroblast. So example for this is you would be able to have your epiglottis, and then you would be able to get your pinna. So these are the two examples for elastic cartilage. Okay, so next important thing is what are the identification feature for an elastic cartilage. The same thing you have to write like that of your hyaline cartilage. So here also you would be able to have the pericardium, which has the chondroblast. You have your matrix, cell extracellular matrix. You would be able to have the chondrocyte, but you should not write cell nesting here. Okay, the chondrocytes are present individually. Okay, you have to write like that. The chondrocytes are present individually. Lacuna is present. Okay, lacuna is present. Okay, so like that you have to write. So here you should not write the, and additionally you have to write another point, presence of elastic fiber. Okay, presence of elastic fiber. So these are the identification feature, justification feature for the slide, elastic cartilage. Okay, so next, so this is your elastic cartilage. So here you would be able to see, so this is your pericardium. This one is your pericardium, the pericardium. So here you would be able to get, this is your elastic cartilage. Okay, this one is your elastic cartilage where the cells are present individually. Okay, the cells are present individually. So here also you would be able to get the ground substances, and you would be able to get the matrix, ground substances or matrix, and here you would be able to get your fibers, elastic fibers. Okay, so next you would be able to see the white fibrocartilage. So what is this white fibrocartilage? So this is the same thing like that of the other two. This is the last one. So here what you are going to see is the cells are arranged in rows. The chondrocytes here, they are arranged in rows, and the fibers here present are type 4 collagen fibers. Okay, you would be able to have the type 4 collagen fibers. Okay, so here you would be able to get the collagen fibers, and the cells are arranged in rows. Okay, here also you don't have any perichondrium and all. In the white fibrocartilage, you are not going to have any perichondrium. Okay, so these are the differences. So here example for the fibrocartilage is, you would be you can tell the costal cartilage, you can tell your intervertebral disc. Okay, so these are the two types of structure that are going to be present in the white fibrocartilage. So you'll be able to have the fibrocartilage. Example are your intervertebral disc and costal cartilage. Okay, so this is the histological diagram of your white fibrocartilage, where you would be able to get the cells are arranged in rows. Okay, the cells are arranged in rows. The fibers and the collagen fibers, they are going to run in different direction. Okay, towards another direction. Okay, so coming towards the differentiation, how to differentiate a hyaline cartilage, fibrous cartilage, and an elastic cartilage. So here, in case of hyaline cartilage, you would be in all the thing, you would be able to get the chondrocyte. Chondrocytes in common in hyaline, fibrocartilage, and elastic cartilage. In all the thing, you would be able to have the cartilage. Okay, so here the thing is, in the hyaline cartilage, you won't be able to see the fibers, but fibers are present. Why you are able to not see? It means refractive index are same. That is the thing. In case of fibrocartilage, the cells are arranged in rows. Okay, and the cells are, and the collagen fibers present in the matrix are arranged parallelly. Okay, in case of elastic cartilage, you would be able to get the chondrocytes are placed individually, okay, and they are been, and you will be able to get the elastic fibers in the matrix, okay. So these are the differentiation between the hyaline cartilage, fibrocartilage, and elastic cartilage. The hyaline cartilage example is your trachea. Fibrocartilage, you will be able to get your costal cartilage and your intervertebral disc. Elastic cartilage, you would be able to get your pinna, and then you would be able to get your epiglottis, okay. So these are the structures you would be able to get here, okay. So with this I conclude the class. So thank you.